Okay, next we're gonna hear an interesting talk from Boaz, which I actually know for many, many years. We did our PhD together, so that should be interesting to see what happened since then. And Boaz is going to talk to us about altered myelination as a novel pathophysiological approach in Williams syndrome.
Okay, good afternoon, everybody. So my lab, we're focused on social behavior abnormalities, and as you can appreciate in this figure, you can see that the multiple brain regions that are active during social behavior in humans, the number of brain regions is very high, and the complexity to study social behavior is very high because of it. And we specifically do neurogenetics studies. We, in many cases, we know the mutation, we know the pathology in the brain, but we don't know the mechanism that is responsible for the pathophysiological aspects. To study the social behavior abnormalities, we use two opposite syndromes related to social behavior. One is the autism that we all discussed now. Specifically, we study also SHANK3 mutant mice. But on the opposite, we study Williams syndrome, which in this case, those patients have hypersocial behavior. They have increased social behavior, and that's what I'm going to be focused on in this lecture. Williams syndrome is caused by heterozygous deletion that I will show soon. It's about 1 to 20,000 successful births. And the main phenotype is hypersociability. Those patients cannot block themselves from interacting with stranger people. The phenotype is very strong. They have high empathy, and overall they are very cute and nice. In addition, they have neurodevelopmental abnormalities, including facial morphology and increased non-social related anxiety. On the cognitive aspect, they have cognitive impairment, as you can see by lower IQ score, and they have other cardiovascular and endocrinological aspects that affect their life. The cause of Williams syndrome is a heterozygous deletion of a chromosomal region on chromosome 7 in humans, labeled here, and if we zoom in, you can see 25 genes that are heterozygously deleted. One of the two copies is being deleted. Each of these genes in this chromosomal region was mapped for the function, and for example, elastin is responsible for the cardiovascular problems and so on, and GTF2I specifically was found to be related specifically to the hypersociability. So we wanted to be focused on the GTF2I. So we all know that when a gene needs to be transcribed and then translate, it has a promoter, and the transcription factor can bind into it, helping the transcription and the translation of a protein. GTF2I encodes a general transcription factor called TF2I, and because of that, manipulating GTF2I can affect many other genes. So previously it was shown that GTF2I is highly expressed in the brain, all around the brain, and also in all cell types that we tried to find. So overall, it's indeed a general transcription factor, making it hard to study. When people try to do full knockout, the embryo was lethal at E11 due to neural tube disclosure and exencephaly. So we decided to make a GTF2I conditional knockout mouse to study better the mechanism. So what I showed you until now is that Williams syndrome is caused by heterozygous deletion, and specifically the GTF2I deletion leads to the hypersociability, but we don't know what is the mechanism. Using a Cre-lock system, we made a GTF2I conditional knockout mouse, and we crossed it with a Cre line, specifically NEX Cre, that here in red you can see the brain regions where we fully knocked out GTF2I from. It is in brain regions related to social cognition. It is specifically in excitatory neurons, so it's a neural study and cell type specific study that we made. And the excitatory neurons allow us to ask a specific question on the largest population of cell types in the forebrain. And also, those cells have projections to affect subcortical regions. So what I'm going to show you now is those mice that are knocked out from GTF2I in the forebrain excitatory neurons. And you can see here by Western blot that the deletion was sufficient. 80% of the protein levels were reduced in the cortex of the 1-month-old mouse. The leftovers come from non-excitatory neurons. And you can see that the specificity is also high because you see that in the mutant mouse, the levels of TF2I are high in the mutant mouse where you have the inhibitory neurons, but in the excitatory neurons labeled here in red, there is a full knockout. So first we wanted to see if this was sufficient to induce hypersociability. We ran the dyadic social interaction and we measured that our mutant mouse spent significantly longer time in close interaction with the stranger mouse. In the social preference test, a mouse supposed to prefer interact with the stranger over the object, and indeed in wild type that's the case, but in our mutant mice the social preference is much higher, and overall they spend significantly longer time with the stranger mouse compared to the controls. On the opposite, when you look at social aggression, you put two opposite mice, a wild type and a knockout, and you let them interact with each other. The more aggressive will push out the other one and will win. Our mice love peace and social behavior, and that's because they are hypersocial and they lose more because of that. Also, in the anxiety-like behavior, we found significantly higher levels of anxiety in the open field and the zero maze. Also, we characterized neuroanatomical features, and we found significant reduction in the brain weight, the cortical thickness, and the dendritic tree morphology of the mutant mice. But as I told you, the GTF2I encodes a transcription factor, so in order to get deeper and understand the mechanism, the molecular mechanism, we ran RNA-seq on whole cortex of 1-month-old mouse, and what we found is a reduction of dozens of genes. You see here the list of genes that were significantly downregulated in the 1-month-old mutant mouse cortex. And you can see that indeed GTF2I was significantly reduced, but what you can see here in yellow, these are all myelin-related genes that were significantly related, with overall of 70% of the total genes that were significantly reduced in the brain of the mutant mouse related to myelination. And I remind you, we knocked out GTF2I only from excitatory neurons. These genes alterations affected cellular pathways also related to myelination, and overall we were pretty much amazed and we didn't expect to see that. So why are we so excited? We all know that myelin helps the increased conductivity of the signal in an axon when you shoot an action potential. If you have myelin, the conductivity is higher. If you have effect on the myelin, the conductivity and the brain function will be affected. And the way it goes is that a neuron has an axon and oligodendrocyte, which is a different population from the excitatory neurons. These are the cells that make the myelin, which you can see here wrapping the axon of the excitatory neurons. We knocked out GTF2I only from neurons, and the levels in oligodendrocytes are intact. We know it from a single nucleus RNA-seq study that I will not show here. And overall, the oligodendrocyte can wrap up with myelin on the axon, and we can compare it to electrical cable that has the isolation that we all know. But if we affect the isolation, the conductivity and the function will be affected. And that's what we first to show a pathophysiological finding related to myelination in Williams syndrome. So we wanted to start characterize the myelination deficits. We started with cellular properties, looking at the number of oligodendrocytes, those cells that can make the myelin and wrap up the axons. And in 1-month-old mouse, we found significant reduction in the number of myelinating oligodendrocytes. And I don't show here data showing lack of differentiation of oligodendrocyte precursor cells into myelinating oligodendrocytes. But in order to have the proper conductivity, the myelin thickness and structure, the ultrastructure, needs to be properly made. So we looked at the ultrastructure of myelin. We took sections of the brains. We looked at the midline of the corpus callosum, and we made a midline section looking at the axons coming towards you with the myelin wrapping the axons. What you can see here on the upper row, you see the controls, the lower one are the mutant. You see that the percentage of axons that are myelinated is significantly lower, and also the thickness of the myelin, measured by G ratio, the thickness is reduced, as you see on the right panel of the EM image. So overall, the ultrastructure was affected, and also the percentage of axons that were myelinated was significantly lower. So if we know that there is lack of myelinating oligodendrocytes and there is effect on the ultrastructure, we can also see if that affects the physiology. In collaboration with Zhigang He's lab from Harvard, we recorded in a mouse, anesthetized mouse, we stimulated on one hemisphere and measured the time it took the signal to get into the other side of the hemisphere. And what we found is that the latency for the peak to arrive from one side to the other, the latency is significantly higher. It takes longer time for the data to be transferred to the other side. And also the amplitude that you get on the other side is lower in the mutant mouse. Now, if we know that the physiology is affected, we can measure behavior which is specifically related to myelin, and there are heavily myelinated axons that run from the motor cortex into the spinal cord that we can measure their function through fine motor skills that we characterize. In the irregular horizontal ladder walking, you let the mouse go through a ladder with bars in a different space between them. So the mouse needs to properly put the hand in a fine motor skills. It's not a coordination, it's the fine motor skills that allow the function of it. And if you have problems with myelination, you will see deficits in this test. And indeed, in forelimb and hind limb, we see increased levels of error rate when we run the test. This corticospinal tract also is responsible for grip strength. When we measure grip strength, in which you pull the mouse gently and measure the strength it has, we see significant reduction in the grip strength. So with this novel pathophysiological characterization, we were asked to show if we can rescue those deficits. If it's a myelination problem, try to fix conductivity or myelination properties. So first we used 4-aminopyridine, which is a potassium channel blocker that can help with the proper conductivity of the signal. If we give single administration IP to a 1-month-old mouse, you see that the baseline of the peak latency in this corticospinal tract recording, if you don't give the drug, the latency is increased. If you give single administration, we rescue the physiological deficits, also in the peak amplitude, and this was also in the corpus callosum. The latency and amplitude were normalized by blocking the potassium channel blockers. This single administration was sufficient to improve the fine motor skills and normalize the error rate of the forelimb and hind limb in the mutant mouse, and also the social preference index after single administration. But there is another drug that can help. Clemestine can improve the differentiation of oligodendrocyte precursor cells into mature oligodendrocytes and then increase the myelination. We use this one, and this indeed rescued the number of the myelinating oligodendrocytes number. It affected and normalized the myelin thickness and affected the social preference, normalizing it to normal levels. So overall, we show here that an excitatory neuron needs to be wrapped up by a myelin from a myelinating oligodendrocyte, but before that, it needs to be differentiated from oligodendrocyte precursor cells. All of this will affect the social behavior, and social behavior will also affect myelination. So if there is a myelination abnormality, you can potentially give clemestine that will increase the differentiation, and through this will improve the myelin condition and social behavior. Other mechanism, suggested mechanism, is to block potassium channels by 4-aminopyridine and improve the myelin conductivity and social behavior through physiological aspects. But all of the things that I showed you now and the last part of my talk will be related now to human findings. Because all the things I showed you now are related to mice, but we try to help human patients. So we asked from the neurobiobank some human tissue samples from frontal cortex from Williams syndrome patients compared to controls. And these patients have the typical deletion from the chromosomal region. So it's not the games that we do with the Cree-lock system manipulating only one gene. This is the real stuff from Williams syndrome patients. We ran RNA-seq, and what we see are about 150 genes that were significantly downregulated. These are here related to myelination specifically. Those in bold were also shown to be reduced in our mouse model. This affected myelination-related cell pathways, and histologically, we found no significant difference in the oligodendrocyte precursor cells in the human tissue samples, but we found significant reduction in the myelinating oligodendrocyte number, as we show in our mice, significantly reduced intensity of myelin basic protein, and the ultrastructure of the myelin in those patients was significantly thinner, as you can see, by increased G ratio. So to summarize, by manipulating one gene specifically, only in 4 brain excitatory neurons, we were able to affect the neuroanatomical properties, social behavior, anxiety related, and we found novel findings about myelination related to reduced expression of genes, ultrastructure of the myelin, the physiology, and myelin-related behavior. Some of these findings were validated in humans, and we have now 2 potential FDA-approved drugs that we can try on human patients and see if we can see some rescue of the deficits. So this is the first molecular evidence related to myelination deficit in Williams syndrome, and the 2 drugs are FDA-approved, and we now work in collaboration with Professor Doron Gotthelf from Sheba on validating our findings and potentially start clinical trials using those drugs. And we hope that with these data and knowledge, we will be able now to improve our understanding and treatment, not only of Williams syndrome, but also of ALS, MS, and other myelination-related disorders. So I would like to acknowledge my lab members. We're a new lab, and Sari Teingel, the lab manager, and Ariel Nir, a student in my lab, they both helped with this study, and we have girl power in the lab. My lab from my postdoc research at MIT, Guping Fang, our collaborators from Zhigang He Lab in Harvard, and the funding agencies. And thank you.